next 45 to 60 minutes, I'm going to talk more on immunology, exclusively on neutrophils, and when exactly neutrophils undergo dysfunction, and how exactly we can restore. When we restore them, what is the impact on our health? These things I'm going to cover in next 60 minutes. So, okay, thank you. Yeah. We know, according to Bible, the life of human flesh lies in the blood. So it means without blood, there is no life for flesh. So since blood gives life to the flesh, by doing three basic functions. One is transportation. It transports dissolved gases like oxygen, carbon dioxide from one part of the body to another part. In addition to dissolved gases, it transmit or transfer nutrients. In addition to that, it protects the body from invading microorganisms, and it inhibits, or we can say it can reduce the fluid losses through damaged vessels, and it can kill or engulf invading microorganisms. In addition to transportation and protection, it also involves in regulation. It regulates body temperature, and it regulates the levels of electrocytes and electrolytes, and it regulates body pH as well as temperature. So, when we draw blood from a healthy human volunteer, when you spin down the blood, we can get two things. One is cellular part, another one is acellular part. When you take acellular part, it accounts for 55% of total volume. It contains mostly water, electrolytes, as well as coagulation factors, coagulation proteins. To some extent, some amount of platelets. When it comes to acellular part, it accounts around 45%. Out of that, 40% accounts for only RBC. Another 5% accounts for both platelets and WBC. From this, we can say RBC is the major cells present in the blood, followed by platelets. And then, when we compare these two types of cells with neutrophils, neutrophil number is very low. So, when it comes to cellular part, there are three types of blood cells. One is RBC, platelets, and WBC. WBC, we can classify them based on the granular structure into two parts, granulocytes and agranulocytes. Granulocytes, they do contain many granules within their cytosol. Agranulocytes, they don't have this side of granules. Upon their activation, these granulocytes release their granular content to the outside of the cell. So, since my talk basically concentrating on white blood cells, from now onwards I am going to talk more on white blood cells. So, what kind of research we are doing in our lab? Like, we are basically working on blood cells, and we are trying to identify the mechanism, how exactly blood cells undergo dysfunction, and how exactly we can restore their function, and when exactly they are going to die, what are the metabolites kills or induces death to either platelet or WBC or RBC. First and foremost, today's talk, restoration of neutrophil function and prevention of apoptosis amid dysfunctional glutathione redox system. Am I audible to? Yes, okay. So this kind of problem generally we can observe during microbial infection. So the persons who are suffering from dysfunctional neutrophils or low number of neutrophils, they are going to get more number of infections, or they are more prone for infection, as well as they will get sustainable infection. Then in diabetes, there is a common continuous activation of platelets and continuous formation of thrombi. It is common in diabetes. It is because of high glucose induced platelet apoptosis as well as aggregation. We are studying on that aspect. When it comes to hemolytic disorders, 60% of people who are suffering from hemolytic disorders, they will die because of thromboinflammation. This thromboinflammation is because of sustained or continuous activation of platelets by heme, cell-free heme and cell-free hemoglobin. Heme induces continuously, heme continuously activates the platelets, and also meantime it induces ferroptotic death in platelet. This kind of death is known as ferroptosis, iron mediated cell death. It is common in hemolytic disorders. In case of sepsis, there will be elevated levels of cell-free stone in the circulation. When these cell-free stones, they activate the platelets and they induce aggregation of both platelets and RBC. Because of that reason, the person who is suffering from sepsis, he will end up in hypoxia, multi-organ damage, all such things. Finally, death will be the final stage. So we are trying to curb the extracellular or cell-free stone-induced platelet activation and aggregation of RBC, all such things. But today I am going to talk more on restoration of neutrophil functions and prevention of apoptosis amid dysfunctional glutathione redox system. So we know immunity. Immunity is nothing but a state of body's defense mechanism against invading microorganisms as well as against to the microbial infection. There are two types of, I am not going to talk more on this, everybody knows about this. There are two types of immunity, innate and adaptive immunity. Innate immunity is non-specific in nature and it happens within no time, it get activated within few minutes. There is no specificity towards that. In innate immunity, there are so many cells, especially phagocytes, they will try to engulf the invading microorganisms, thereby they will try to kill the microorganisms. If microorganisms, invading microorganisms, breach the innate immunity, then only adaptive immunity will get activated. It is specific in nature and it takes long time, minimum 96 hours. So in adaptive immunity, immune cells will produce antibodies against specific antigen and they will try to kill. It includes B cells and T cells. So in the innate immunity, there are, as I said earlier, there are so many cells. It includes basophils, eosinophils, neutrophils. All of them are granulocytes, whereas mast cells and natural killer cells are agranulocytes. Among these, I am interested on neutrophils. A neutrophil being a innate immune cell, their number is huge when compared to rest of the granulocytes and agranulocytes. That's why we are interested on neutrophils. So what is neutrophil? Neutrophil is an innate immune cell. It is most abundant granulocytes. Since it is a granulocytes, you can find many number of granules in their cytosol. And it is having multi-lobed nucleus. That's why we can call them, these cells as polymorphonuclear cells. And they are phagocytic in nature. They can engulf the invading microorganisms by phagocytosis. As I said earlier, neutrophil is a granulocyte. It contains 3 different types of granules in addition to one more, 4. Those are azurophilic granules, gelatinous granules, specific granules, as well as secretory vesicles. If you observe the content of these granules, you will come to know these granules contains hydrolytic enzymes in them as well as antimicrobial agents. Antimicrobial agents as well as hydrolytic enzymes are common in their vesicles, in their granules. In addition to these two, you can find chemoattractants like lactoferrin, lipocalin. These are the 2 examples for chemoattractants. I will explain what are chemoattractants, why they are so helpful in neutrophil function. So in addition to that, we have secretory vesicles. They do contain certain membrane receptor as well as plasma proteins. So what is the exact function of neutrophil? So as I said, it is a phagocytic cell. It tries to engulf the bacteria are invading microorganisms since it is a phagocytic cell. So when you consider size of the bacteria is small when compared to neutrophil size, so neutrophil try to engulf the bacteria. This process is known as phagocytosis. If neutrophil is unable to engulf the bacteria, in such situations, what neutrophil does, it undergoes degranulation. Since it is undergoing degranulation because of the bacterial toxins released by bacteria, since this neutrophil is unable to engulf the bacteria, by using bacterial toxin, it undergoes degranulation. Once it undergoes degranulation, in addition to hydrolytic enzymes and antimicrobial agents, chemoattractants will also release. Those are lactoferrin, lactoaderin, and as well as lipocalin. Once they release these chemoattractants, the neutrophils which are present in and around, they will come and play along with existing neutrophil. So all these neutrophil sit upon the bacteria and they undergo degranulation. Since degranular content contains hydrolytic enzymes and antimicrobial agents, totally they will kill the bacteria. So either phagocytosis or by undergoing degranulation, neutrophil try to kill the bacteria. If neutrophil kills the bacteria all alone, then I can call that as a phagocytosis. If neutrophil requires some sort of assistance with other neutrophils, then I can call that as a degranulation process. If these two processes won't work in order to kill the bacteria or invading microorganisms, then the third function, netosis, formation of neutrophil extracellular traps. If these two functions won't work, phagocytosis and degranulation won't work, then only neutrophil undergo netosis. There are two types of netosis, vital netosis and suicidal netosis. Vital netosis happens within 60 minutes of their activation, whereas suicidal netosis requires around 180 minutes. So what it does during netosis means it will eject its own DNA. Decondensed DNA will be ejected and it serves as a trap, spider web, in order to occupy the bacteria. That DNA, ejected DNA, is decorated with hydrolytic enzymes as well as antimicrobial agents. So by using that, it will undergo suicide. It suiciding itself by ejecting its own DNA in order to kill the bacteria. So this kind of netosis is considered as suicidal netosis. So it takes 180 minutes to happen, minimum 120 to 180 minutes to happen. And once after the netosis happens, neutrophil will die. Whereas in case of vital netosis, neutrophil are viable, and meantime they will eject small amount of DNA in order to entrap the bacteria. So these two kinds of netosis happens. Now I would say, if neutrophil all alone engulf the bacteria, then phagocytosis. If neutrophil requires assistance from other neutrophils, I can say degranulation. If neutrophil kill by itself in order to kill the bacteria, then I can call that as a netosis. By using these three different functions, your neutrophil serve as an innate immune cell in order to kill the invading microorganisms. Okay. Okay. So in our lab we asked the question: is there any crosstalk or is there any role for redox system in neutrophil function? Whether redox system play a crucial role in phagocytosis, degranulation or netosis. We asked a simple question like, everybody knows about glutathione. Glutathione system is a very well known redox system in every cell. Every cell is having glutathione in a millimolar level. When the cytosolic concentration of glutathione is in millimolar level, whereas extracellular concentration of glutathione is micromolar. It means 1000-fold difference in the concentration of glutathione in the cytosol when compared to outside the cell. Okay. So when glutathione cycles, this is the structure of glutathione. It is a tripeptide having glutamic acid, cysteine and glycine, and this the peptide bond between cysteine and glycine is different. It is an unusual peptide bond. So this is the glutathione cycle. When in order to quench the radical, in order to reduce the radical, your reduced glutathione undergo oxidation. It becomes oxidized glutathione. This oxidized glutathione is so reactive and it binds to, it interact with another active oxidative glutathione and form a GSSG. Okay, and this enzyme is mediate this reaction, glutathione peroxidase. It is a selenium dependent enzyme. This enzyme convert superoxides like H2O2 into water. Whereas this oxidized glutathione replenish into converted into reduced glutathione by the enzyme glutathione reductase. It requires NADPH as a cofactor or reducing power. It comes from HMP shunt product. So this is the cycle of glutathione. Like that, glutathione always try to convert from reduced to oxidized in order to quench the free radical, and oxidized glutathione to reduced glutathione in order to replenish the reduced glutathione in the cell. So in healthy state, always the glutathione concentration is more when compared to oxidized glutathione. During disease state or unhealthy condition, more amount of reduced glutathione will be converted into oxidized glutathione in order to quench the radical. So the ratio between reduced glutathione to oxidized glutathione will vary. It depicts whether cell is active or not. Okay, as I said earlier, always the reduced glutathione level is so high when compared to oxidized glutathione. That's why ratio will vary from 30:1 to 100:1. So now, from this I can say, in order to have a normal function, every cell should have more amount of reduced glutathione when compared to oxidized glutathione. If something happens to reduced glutathione levels, it leads to malfunctioning of any cell. Okay, I am taking this as an example because this glutathione is a major redox system in every cell. In addition to that, we have glutaredoxin, thioredoxin, peroxyredoxin redox systems, as well as bilirubin, biliverdin cycle. This will assist the glutathione cycle. So based on this, physiologists are telling glutathione is considered as mother of all antioxidants, the master of detoxifier, and master of immune system. So from this I can say there is a crosstalk between glutathione level and neutrophil function. Okay, based on this question or hypothesis, we searched many literature. We did lot of literature survey and we found some interesting papers related to this. So first paper described about melioidosis. Melioidosis is a clinical condition. Generally you can observe melioidosis in diabetic patients. That too, diabetic patients who are living in coastal area. Now you won't find this much, many number of melioidic patients in mainland area. Generally, the diabetic patients who live in coastal area, you can find lot of melioidosis. Melioidosis is a clinical condition where you can find infection by Burkholderia pseudomyeli. Burkholderia pseudomyeli is a bacteria. It infects diabetic patients because there will be a dysfunction of neutrophil. So because of that reason, invaded Burkholderia pseudomyeli cannot be phagocytosized by neutrophil. So because of that reason, you will end up in infection. So they dissected. They shown there is a huge difference between neutrophils from healthy and diabetic patients with respect to response towards Burkholderia pseudomyeli. So from this they observed that the neutrophil from healthy condition readily phagocytosize the exposed Burkholderia pseudomyeli, whereas neutrophil from diabetic patients, they could not able to engulf the pseudomyeli bacteria. They observed and they published the paper in immunity, infection and immunity. Yan et al. He showed that glutathione reductase play a very crucial role in neutrophil function. because glutathione reductase only convert oxidized glutathione to reduced glutathione. I said already, for neutrophil function there should be always increased levels of glutathione, reduced glutathione when compared to oxidized glutathione. Then same group, Ann et al. He showed glutathione reductase is essential for host defense against bacterial infection. Then very recent, in another work, Alain et al., he showed role of myeloperoxidase and bacterial metabolism in chemiluminescence of granulocytes from patient with chronic granulomatous disease. Chronic granulomatous disease is another genetic disorder. The people who are suffering from CGD, in them there is a defect in NOx enzyme, NADPH oxidase enzyme. When you take a cell, there are two places where exactly you can find generation of RAS. One is mitochondria, another one is cytoplasm. The major source is mitochondria only, because since electrons are moving through ETC, there will be a continuous dissipation of free radicals. There are two enzymes, defensive enzymes like superoxide dismutase. In spite of that also, if you inhibit the ETC, there will be a continuous leakage of radical. So we can say mitochondria is the source of radical generation. In cytosol, for example, if you take macrophages and neutrophils, they are phagocytic cells. Since they are phagocytic cells, they will try to engulf the bacteria. Once they engulf the bacteria, they should kill the bacteria. For killing the bacteria, they require superoxides or free radicals. Those free radicals will be generated by NOx enzyme, NADPH oxidase enzyme. It is a membrane bound enzyme. It uses NADP and NADPH as substrate and generates superoxides. Those superoxides will try to kill the bacteria. This entire process is known as oxidative burst. It happens in neutrophil as well as macrophages. By using oxidative burst system, they will try to kill the engulfed bacteria. Okay? So, in this CGD disease, there is a defect in NADPH oxidase. Since there is a defect in NADPH oxidase, the neutrophils and macrophages of those patients who are suffering from CGD, they will engulf the bacteria, but they could not be able to kill the bacteria. So that's why these patients are prone for sustained infections. Okay, we from this background, we came to know that there is a crosstalk or there is a connection between redox system and functionality of phagocytic cells. So these are some pictures of the patients who are suffering from myelodiosis. These are wounds you can find all over the body. This kind of myelodiosis is common in diabetic patients who are living in coastal areas when compared to mainland area. So these are the infants who have CGD. They are suffering from chronic granulomatous disease. So from this we can say myelodiosis is common in diabetic patients. We can say there is a decreased levels of reduced glutathione over there, and because of this reason they are unable to phagocyte the bacteria. When it comes to CGD, chronic granulomatous disease, it is a genetic disorder. Because of that reason we can expect defect in the NOx enzyme, NADPH oxidase enzyme. So because of this reason, neutrophil exhibits normal phagocytic function, but they are unable to kill the engulfed bacteria. So because of that reason, these patients are vulnerable for sustainable infection. So from this background, we can say a tightly regulated balance between RAS production and reduced and oxidized glutathione cycle is in neutrophil is critical for an efficient microbicidal and phagocytic efficacy. Can we say like this? So the main thing is we need to have more amount of reduced glutathione all the time in order to have better phagocytic effect as well as antimicrobial property of neutrophils. The strategic use of molecules that can protect the cells as well as enhance the antimicrobial competence of neutrophil during clinical condition is must or is required. That gives more meaningful therapy. The usage of non-specific and low molecular weight antioxidants at high doses have failed in the prevention or mitigation of several pathologies. But usage of redox modulators targeting specific ROS-mediated signaling pathways may be highly effective. There is a huge difference between redox modulator and antioxidant. If you say antioxidant, the purpose of antioxidant is to quench the radical. That's all. Whereas redox modulator not. They will quench the radical in order to maintain overall hemostasis of free radical antioxidant as well as pro-oxidant system. They will maintain overall hemostasis of both pro-oxidant and antioxidant system. So, and also meantime, we did literature survey and found there are so many papers related to GPX mimetics. GPX mimetics means glutathione peroxidase mimetics. Glutathione peroxidase, the enzyme which is very essential in order to quench the radical by converting reduced glutathione to oxidized glutathione. But no study, we didn't find any study related to GR mimetics. If I ask chemistry people, they will say since GPX is having selenium in it, so it is very easy to make GPX mimetic. Whereas in GR, it is FAD containing enzyme and NADPH cofactor. Because of that reason, we cannot. Because FAD is not involved in oxidation reduction. Whereas selenium is involving oxidation reduction. It is easy for them to make GPX mimetics rather than GR mimetic. So based on all these background, we make an hypothesis. We selected melatonin as a redox modulator. Why we have selected melatonin is it is clinically approved and it is produced in your body only. So it maintains sleep and wake cycle, or it maintains biological circadian rhythm. Okay. Most of the accidents happens between 3:30 to 4:30 in the morning. Most of the it is all because of this hormone only, pineal gland, which is present just in the stem of brain. It releases maximum amount of melatonin between 3:30 to 4:30 and induces lot of sleep in humans. So because of that reason, normally accident happens between that time. And also when you drink more amount of buttermilk, when you eat banana, when you eat lot of rice, it induces sleep. Because banana, buttermilk and rice, they do contain lot of melatonin. So that induces this one. So it is clinically approved and easy to use and nothing will happen. So because based on this background, we selected melatonin. Melatonin serves as a redox modulator, or we can call melatonin as a conditional antioxidant. We are not calling antioxidant, we can call conditional antioxidant. So this is the structure of melatonin. It is a derivative of tryptophan. It is nothing but N-tryptamine. Okay. So melatonin works at 3 levels. It works at membrane level, plasma membrane level, it works at cytosol level, and as well as it works at nuclear level. So at membrane level, there are 2 receptors, MLT1 and MLT2. By binding to those receptors, it induces lot of signaling cascade within the cytosol and serve as a redox modulator. And it slowly diffused into the plasma membrane also. Then in cytosol, it directly quenches the radical. It serves as antioxidant. In nucleus, it enhances. It acts as a transcription activator. It activates NRF2 transcription factor. NRF2 is a transcription factor. It activates gene encoded for antioxidant enzyme machinery. So thereby, it works as a redox modulator. So that's why we selected melatonin as a redox modulator in order to prove our hypothesis. So, what are the components we have used in order to prove our hypothesis? First one, before that we asked a question, can melatonin restore the neutrophil function? If so, can we use melatonin as a adjunctive therapy in addition to existing therapy in order to treat microbial infection. That was our motto. So we used melatonin as a conditional antioxidant or redox modulator, and we used 2-AAPA. This chemical, it is a big name for 2-AAPA. Shortly we can call that as a 2-AAPA. This chemical can cross the plasma membrane and target glutathione reductase enzymes in the cytosol. By targeting the glutathione, if you target glutathione reductase enzyme, that enzyme cannot convert oxidized glutathione to reduced glutathione. So now cell will have very low amount of reduced glutathione and very high amount of oxidized glutathione. You know that. That's why we use 2-APA. It specifically cross the plasma membrane. And we use E. coli in order to confirm phagocytosis. Then we use LPS. It is an activator of degranulation. When neutrophils are exposed to LPS, neutrophils readily undergo degranulation. Then we use neutrophil degranulation markers in order to confirm whether neutrophil undergone degranulation or not. It is very difficult to estimate the antimicrobial agents as well as chemoattractants and hydrolytic enzymes. Instead of that, if we use CD markers, it will be easy for us to know whether neutrophil undergone degranulation or not. So we used PMA in order to induce netosis. So we use 3 different things to confirm phagocytosis, degranulation and netosis. So very first question, we have to use two set of cells. One is normal neutrophils. In those, glutathione cycle is perfect. In another, we use 2-APA in order to block the glutathione reductase. So we can call them as a GR deficient neutrophils, glutathione reductase deficient neutrophils. In such neutrophils, you can expect very low amount of reduced glutathione and very high amount of oxidized glutathione. You know, these 2 types of cells we have used. So once we use neutrophil, normal neutrophil and GR deficient neutrophils, these cells were exposed to H2O2 and we induced oxidative stress by giving superoxides. And we observed apoptotic markers as well as reactive oxygen species, and we found elevated levels of reactive oxygen species and apoptotic markers. Now I can say H2O2 readily induces death to GR deficient neutrophils. When these cells were initially treated with melatonin followed by H2O2, then we assessed levels of ROS and apoptotic markers, and we found significant decrease in the ROS, reactive oxygen species, and cells were viable. Cells, they did not undergo death. From this I can say melatonin protect GR deficient neutrophils from oxidative stress induced death. In order to confirm that, we did cell viability and as well as pH externalization as the eat me signal. Okay? So then we asked the question, can melatonin restore phagocytic property of neutrophil or not? For that also we used 2 set of cells, neutrophil, normal neutrophil and GR deficient neutrophil. GR deficient neutrophils as well as normal neutrophils were exposed to E. coli cell. These normal neutrophils readily engulf these bacteria, whereas GR deficient cells were unable to engulf the bacteria. Okay, in another set, same cells were initially treated with melatonin, then exposed to bacteria. Those cells were readily engulf the E. coli cells. From this I can say melatonin restores the phagocytic property of neutrophils. Same concept we have used and exposed to LPS. These GR deficient cells were unable to express CD63 and CD35. These 2 CDs get expressed more when neutrophil undergo degranulation. When these cells were initially treated with melatonin followed by LPS exposure, these cells were readily expressed CD35 and CD63. So this also clearly suggests that melatonin restores the degranulation property of neutrophils. Then followed same set of experiment we did for netosis. Here we use LPS or PMA as a netosis inducer. Here also GR deficient neutrophils, they were initially treated with melatonin followed by exposure to LPS or PMA. Those cells were readily undergone netosis. Whereas only GR deficient cells, when they exposed to LPS or PMA, they did not undergo netosis. From this data clearly suggests melatonin is restored the functions of neutrophils. Okay. When you see this picture, initially we thought when GR deficient neutrophils get exposed to LPS or PMA, we observed there should be elevated levels of ROS. Maybe we thought like 10 or 12 fold increase in the ROS, but we found very low amount of ROS when compared to this. This made us to think a big, and this made us to identify molecular mechanism. So now from this data I can say for neutrophil, in order to do its function, there should be a essential amount of ROS. Got my point? Minimal amount of ROS is required for neutrophil to do its function. If the minimal ROS level goes down, it cannot do its function. Got my point? Okay, if you give antioxidants in that situation, it can quench all the radicals. Instead of antioxidant, if you provide a redox modulator, it maintains the minimal ROS. Thereby it restores the functions of neutrophils. That was the concept, okay, from this. Okay. Okay, now reviewers will ask, so you are showing some things, final. You are showing final end product, and you are not telling what is the mechanism behind it. How exactly your melatonin restoring the neutrophil function? How can you say? You are showing end result, that's all. No one cares. So in order to prove that point, what we have done, we did lot of literature survey and we found some points. There are two things is essential for neutrophil to do its job. One is NOx enzyme. Everybody knows now. NADPH oxidase. It is very essential for the generation of superoxides. Those superoxides are very essential for killing the engulfed bacteria. You know that. And also meantime, those superoxides are essential in order to do its normal function. I said for neutrophil, there should be a minimal ROS to do its function. That is maintained by NOx enzyme. Then another one, actin dynamics. You know, actin cytoskeleton, it is present just beneath the plasma membrane. It only decides size or shape of the neutrophil. So these two play a crucial role for neutrophil to undergo degranulation, to do phagocytic job, to undergo netosis. Okay, this is the completely activated complex of NOx enzyme, NADPH oxidase. It is a multimeric protein. It uses NADPH as a substrate in order to generate superoxides. Okay. Now we came to know that there could be two possible targets. One is NOx enzyme, another one is actin dynamics. Now we asked a question: what is the role of actin dynamics and what is the levels of NOx enzyme in normal neutrophils as well as in GR deficient neutrophils? And we asked simultaneously we asked a question whether melatonin can restore the NOx enzyme and actin dynamics. So for that, what we have done, we used two set of cells, normal neutrophil and GR deficient neutrophils. From them we assess the levels of ROS in presence of two individuals, LPS and PMA, and also meantime we assess the levels of NOx enzyme activity. And we found there is a significant restoration of NOx activity by melatonin. Okay? Then, when exactly the enzyme loses its function? There is a question. So, what happens when in nature everything will be, once it generates or once it gets inside, it has to do its function. After its function, it should eliminate from the system in order to accommodate newer one. That is the thing. Synthesis, function and elimination or degradation. This is the process. Same thing is true in case of enzymes. When enzymes get defected, it will undergo degradation by proteasome. Okay. Before that, if system wants to defect the enzyme, there are two ways. Glutathione, normal way. Normally, protein undergo modification. Once protein enzyme undergo glutathione, for example, glutathione or simulation. There are so many modifications are there. If once protein undergo glutathione, it may be targeted to proteasome. It will target it to proteasome. In any cell, if there is a lot of oxidative stress, in normal case, after its function, generally there are enzymatic machinery. By using that enzymatic machinery, glutathionylation happens. Once certain protein get glutathionated, it will be targeted to proteasome. That is in normal case. Whereas in unusual cases, or we can say during pathologies and all, the oxidative stress level increases when cell is undergoing death. During such situation, you can expect elevated levels of oxidized glutathione when compared to reduced glutathione. When oxidized glutathione level get increases, there will be a non-enzymatic way of glutathionylation of targeted proteins. So those proteins will be targeted to proteasome. Thereby, cell will die. So in order to confirm that, first we wanted to know whether cytosolic proteins are undergoing glutathionylation or not, or glutathionylated or not. For that, we did blood by using anti-GSH antibodies. See, you can see there is, in normal case, there are roughly 1 or maximum 2 proteins which are getting glutathionylated. When we treat with AAPA, we can consider this as a GR deficient neutrophils. You can find there are 5 to 7 proteins which undergone glutathionyl. In case of LPS, there is also some more. In case of LPS exposed to GR deficient neutrophils, the levels get increases when compared to these 3 lines. Yes or no? In case of melatonin, the levels get restored back. So now I would say melatonin is blocking glutathionylation of essential enzymes, essential proteins for neutrophil function. Can I say like that? So I don't know which one, but from this I can say melatonin is blocking or inhibiting glutathionylation of essential proteins or enzymes which are required for normal neutrophil function. Can I say like that? Yeah. So same thing is true in case of actin dynamics. When oxidized glutathione level get increases, it leads to glutathionylation of actin. This was published in Immunity as well as JCV in earlier years, like 2017 and 2012. Beyond doubt, they proved that when there is a oxidative stress in cell, during such situation, actin dynamics, actin proteins are getting glutathionolated. Now I would say our target is nice. Both NOx and glutathione, actin proteins are undergoing glutathionylation in GR deficient neutrophils. So this could be the possible reason for dysfunctioning of neutrophils. My question is whether melatonin can block the glutathionylation of actin proteins and NOx enzyme. That was our question. So in order to prove that point, we did IP, immunoprecipitation. When we did IP, we came to know that yes, melatonin is blocking glutathione of NOx enzyme as well as glutathione of active dynamics. So by doing that, it is restoring the functioning of neutrophils. Okay, we did IP and all. So from this, I can say glutathione reductase is very essential for neutrophil function. First point, melatonin effectively restores ROS generation, phagocytosis, net formation in GSH and GR activity deficient neutrophils. We use GSRs also. Parallelly, we block the GR synthesis by using BSO as a chemical. It inhibits glutathione synthesis in the cell itself. Thereby also you can deplete the reduced glutathione level. Either by inhibiting this enzyme or by inhibiting the enzymes which are involved in glutathione synthesis, you can create glutathione deficient cells. We have showed an uncharted property of melatonin that modulates the redox status and aid to execute neutrophil function under cellular GSH depletion and GR activity deficiency. Melatonin could be an appropriate candidate for auxiliary therapy to treat microbial infection, including viral infections, even under chronic disease conditions. Okay, this is the schematic representation of exactly melatonin restores the neutrophil function. So, the entire data was published in Journal of Pineal Research. It is a high impact journal, and we submitted this paper on April 7th, 2020. First national lockdown happens in 15th March or 20th March 2020. During lockdown only we did all the experiments and we wrote the paper. And we submitted that journal and it got reviewed and we received comments by June 1st and June 8th we submitted and it got accepted in 21st June. Okay, so in this paper we mentioned about COVID-19 also, and we said we can use melatonin as a adjunctive therapy in order to treat COVID-19 and all, and we wrote discussion and in introduction also we wrote everything. But editor said you never used any sort of clinical samples related to COVID-19. That's why you cannot use the word COVID-19 in this paper. And he made a strict this one. You cannot use COVID-19. So you can use in a conclusion part. It can be extrapolated to COVID-19. That's all. In title or introduction as well as discussion, you should not use any sort of this one. But once we published this paper, so many other researchers, they followed our work and they came up with like this clinical trial to test the efficacy of melatonin in COVID-19. They did, and it was effective. Then followed is melatonin deficiency is unifying pathomechanism of high risk patients with COVID-19. They said whether melatonin levels were low in patients who are suffering from severe COVID-19 infection and all. Okay, with this I thank my mentor, Professor K. Kemparaju sir, for his continuous help and support during my entire study, and I thank my students Naveen Kumar, Dr. Naveen Kumar, Dr. M. Shekhar and Dr. Manikanta. I thank Professor Razal J. Reiter as a source of inspiration. He worked on melatonin. He is still working on melatonin since from 45 years. He is a founder editor of Journal of Pineal Research. He got retired in 2017. He got retired. Still is working. He is a retired professor. I will consider him as a source of inspiration. And I thank funding agency, BST sir. Thank you for patient hearing. If you have any questions, I am happy to answer. Thank you.
Thank you so much, sir, for your very informative talk.
Thank you.
Any questions or queries regarding this?
Yeah, if you have any
Questions
clarifications
from audience?
melatonin is available in medical shops now.
Yeah.
Sir, I have one doubt.
Yeah, please.
So how melatonin can
Wait,
reduce
wait,
apoptotic?
I'm sorry.
How melatonin reduces apoptotic markers?
How melatonin? Yeah, okay. Apoptotic markers are cell death markers, I can say. If cell is under, there are 10 different types of cell death. We have apoptosis, necrosis, autophagy, ferroptosis, pyroptosis, paraptosis. Like there are so many. Well-studied mechanisms are 4 or 5. So in apoptosis there will be two things, intrinsic and extrinsic pathway. If mitochondria get damaged, because of that reason, when mitochondria get damaged, ETC will get collapsed. So RAS level get increases like anything. Okay, so because of that reason, MPTP formation or pro-apoptotic BCL2 proteins will come together and form a pore. Because of that reason, cytochrome C will come out of the mitochondria and form an apoptosome, and it activates caspase 3. Then pH sectionalization happens, and also meantime there will be a, when calcium level get increases in the cytosol, calpain enzyme will get activated. It degrade actin dynamics. Because of that reason, entire structure of cell, cytoskeletal structure get destroyed. There are so many events, we can say, multi-factors are involved in apoptotic pathway. Whereas melatonin, by serving as a conditional antioxidant, first point, it works, and by activating cellular signaling, it prevents apoptosis. By activating cellular signaling. So it brings down intracellular calcium level. It won't allow endoplasmic reticulum to release calcium to the cytosol. So by doing that, first point, and also by quenching the radical and by increasing the mitochondrial health, it can prevent apoptosis. We cannot show all these things in our paper. So we will take 2-3 markers and we show whether it is working or not. So these are the mechanisms. I said everything, but I cannot do all these things. So in this way, melatonin can prevent apoptosis.
Thank you, sir.
Thank you.